Okay, so let's go over what you need to understand about the 2B, as I call it, in the mitochondria, the cycle that's involved. We're going to call it the citric acid cycle. That's named that because the first molecule made in the cycle is citric acid or citrate. It's the same thing, just depends if the hydrogen ion is attached or not, or the hydrogen atom. So we'll call it the citric acid cycle. It has been called the Krebs cycle. I prefer you not to call it that. I mean, it just seems modern day they tend to call it citric acid, or the tricarboxylic acid cycle. So you'd see it as TCA cycle. Sometimes you see that. It's all the same. We'll call it citric acid. So this is the extent you need to know about it, and the ideas of why things are happening, what it goes in, what comes out, what gets transformed, and just remember all the steps are enzyme catalyzed. I don't need you to know any enzyme names for our purposes, but they don't just randomly happen, and they're not literally in a circle either. The circle is just to show that the starting molecule is your end molecule, but these reactions just happen in the matrix of the mitochondria, but not literally in a circle. All right, so here's our starter. And again, I'm drawing this like in the other sections very schematically, but it's a 4-carbon compound. Just like any cycle, there's a starter molecule, which is going to be the ending molecule. This one's name is oxaloacetate. I'm not too concerned about that, but there you go. And when it happens, we're going to take that acetyl-CoA that was made and it's unstable, reactive from the oxidation of pyruvate, and add that, this is the whole point, bump off the CoA and put those 2 carbons onto this 4-carbon compound. There's your 4 carbons, and then you're going to add 2 more carbons. Great. So now we have another compound. This is actually the citric acid. Okay, so we've got our carbons into the cycle. Why is that important to add carbons? Because that's where we're extracting all the electrons. So you add the carbons in, and then you extract all the electrons from them. That's the whole point of this. Why is the point of citric acid cycle? Let's say extract electrons from carbon compound. So let's start. Remember, in order to extract electrons, the key here is you break a bond. So let's just remember this. Break a bond. Extract electrons. Key idea. So we want to extract some electrons. Great, let's break a bond. So we'll break a bond. What does that mean? That means this carbon comes off, and it's going to leave in the form of CO2. And that means that we can extract electrons. So when we extract electrons, they have to go into an empty electron carrying molecule. So in the case of cellular respiration, that is your NAD plus. The plus sign is indicating again that it is empty here, like meaning that it doesn't have those electrons in it. If you know about taxi cabs, if the light is on, it means that you can hail it because there's no people in it. If the light is off, it means that it's full. So you can think of the plus as the light on. Without the electrons, that's also a way. Electrons are negative, so the plus means it's without that negative. Okay, so we add the electrons to that, extracting them after during that bond breaking, and that makes your NADH, which is with the electrons, and that is going to go to the electron transport chain. That's the whole point here. Great. Still have some carbons left. And I'll show you this. Here is your NAD plus and your NADH, what it goes to here. So it's a relatively large molecule. It's a coenzyme. We usually just call it NAD plus NADH though. So you can see it has some, its name, nicotinamide adenine dinucleotide. And so we see here, this is the nicotinamide group, dinucleotide, and these are adenine. So just like the A in DNA and RNA type, this is like a RNA type because it has two OH groups there. But so it's like the same type of molecule as you'd have in RNA, but it's attached together, and then nicotinamide's different. So in any case, this is the plus version, meaning that it doesn't have those electrons. You can see when it picks up the electrons and hydrogens, then you have that added, which means that you don't have the positive charge because you've picked up those electrons. And so this is the, quote, reduced form, because reduction, like this NAD plus is reduced to NADH because it gains electrons. That's the rig part, right? Reduction means gain. And then once it loses those electrons by donating them to another molecule, it's going to be oxidized. So you would say that NADH is oxidized back to NAD+. And so you just have this cycling here within the mitochondrion. All right. So that's how NAD plus NADH works. Very similar to the NAD. This is a photosynthesis thing. Remember, we have NADP plus and NADPH. So the P just means phosphate group, but you can also think of the P as in the one in photosynthesis. Let's extract some more electrons. So what are we going to do? We're going to break a bond, extract electrons. So we broke the bond, which means we released the carbon molecule. In this case, CO2 is the molecule. And we extracted electrons. What do? We bring in the empty carrier, the NAD+. It picks up electrons and leaves at NADH. And as always, it goes to the electron transport chain. Those electrons are going, into the NAD+. Okay. Then there's some fancy stuff that goes on here. I'm not worried about it. What I need you to know is that there's some steps. Part of those steps is making a little ATP. To make ATP, we're going to have ADP, and it's going to come out as ATP. Draw that, leave a little room up there, just a little. And the way this happens is not with ATP synthase, the enzyme. It's going to happen through the substrate level phosphorylation. This is the same as what happens in glycolysis. So to remind you here, or if you're looking at this the first time, substrate level phosphorylation, basically you have the substrate, which is a molecule which is going to be bound by the enzyme, and there's your phosphate attached to that substrate, and then you have ADP as your second substrate, really, because it's bound to the enzyme. And then a reaction occurs where you have the phosphate group get transferred. You can see the phosphate group got transferred onto the ADP, which is now ATP, because di means 2 phosphate groups, tri means 3 phosphate groups. Where's the third one here? There's your 1, so 1 plus 2 equals 3. And that is your substrate level phosphorylation. So I guess we'll draw it like this. Let's say, so phosphate does come into the system, and there's a substrate that gets that phosphate attached to it. I'm just going to make it a triangle. Okay. And in any case, that phosphate group gets bumped off of there. There's the triangle to pick up more phosphates. So then we have some more steps here, steps. And part of that rearranging to get back to the starter molecule, the oxaloacetate. We don't break off any more carbons, but we do extract some more electrons by breaking bonds to rearrange. So one of those extractions is a related molecule to NADP or NAD+, so FAD. So FAD can pick up some electrons too, or as well, 2 of them, and it ends up being FADH2. So that is, again, going to carry electrons to the electron transport chain, and then we make another round of NAD plus to NADH. Again, has the electrons, and guess where it goes? electron transport chain, plus 2 electrons in there. Okay, and then we're back. The molecule has been rearranged back to this starter oxaloacetate here, and then it can happen again. I don't need you to track the carbons in particular. This is what I need you to understand here. Break a bond, extract electrons. When that bond's broken, you're releasing CO2 as a byproduct. CO2 can go diffuse out of the mitochondrion and out of the cell. Could go and be used in photosynthesis. Eventually, that's what would happen. It may go to the atmosphere and then be brought in again, but that is the carbon compound, right? That's part of that cycle, the carbon compound breakdown. So we say carbon compound to CO2, that's what this is, carbon compound to CO2. And that's a citric acid cycle.